Good day everyone. Today, I will be explaining the process of making a herbarium. First, let us understand what a herbarium is. A herbarium is a systematically organized collection of plant specimens that are collected, pressed, dried, and permanently preserved for scientific study. Each specimen is carefully mounted on a sheet and labeled with important information. Herbarium helps to support plant identification, ecological research, and the documentation of biodiversity over time. It also enables researchers to examine real plant structures even long after collection. Herbarium is important in both education and scientific research. Firstly, it helps in plant identification. By comparing specimens with existing collections, we can accurately identify plant species based on their physical characteristics. Secondly, herbarium acts as a reference for research. It provides reliable and preserved samples for study in botany, taxonomy, and ecology. Thirdly, it functions as a historical record. Scientists can study changes in plant distribution, structure, or characteristics over time. Lastly, herbarium supports environmental monitoring. It helps researchers to track environmental changes such as climate change, habitat loss, and biodiversity decline. There are 7 main steps in preparing a herbarium specimen. Firstly, choosing, then the second one, collecting, then move on to pressing, drying, identifying and labeling, mounting, and the last one is specimen care. Now, I will explain each step in detail. The first step of producing a herbarium starts from choosing the specimen. At this stage, we must carefully select a plant which is fresh, not damaged, and free from disease or insects. Besides that, we need to choose a plant that shows the typical features of the species. Not only that, the specimen that we choose should fit well on a herbarium sheet and be easy to press. This means that the plant should be small enough or can be arranged in a way that fits neatly on a sheet. And lastly, we need to avoid rare or protected plants. In fact, we can collect from common species. Then, the step 2 is collecting the specimen. Here are some collection techniques that we need to follow. First, we cannot collect rare or endangered species, as this can negatively impact the biodiversity and may also be against environmental regulations. Besides, only a small and sufficient sample of the plant should be collected. Over-collecting must be avoided to ensure that the plant population in the area is not reduced. Thirdly, we should also avoid harming surrounding plants, soil, or ecosystem during the collection process. Not only that, we need to use proper tools such as scissors or pruning shears to cut the specimen carefully. And last but not least, we should ensure that the plant collection does not affect future growth or biodiversity. These are the materials that needed for specimen collection. The first one is pruning shears or scissors. Second, gardening gloves. Third, plastic bags. And the last one is mud foam. These tools help to ensure the collection process is efficient, safe, and organized.
First step of creating a herbarium starts from specimen collection. For this project, I chose ornamental plants that can commonly found in gardens and also public areas. I chose plants with distinct leaf margins so that their features can be clearly observed. Before I start my specimen collection, I will wear my protective gloves. So wearing gloves is very important because some plants may consist of thorns or irritating substances that may lead to skin irritation and allergic reactions. I wear gloves is also to avoid from scratches, insect bites, and also plants with plant sap.
During collection, I use appropriate tools which is scissors to cut the plant parts carefully. I collected parts that clearly shows the plant's important structure, which is the leaves. Each specimen was then placed carefully into a plastic bag or folder to prevent damage before preservation. Before sealing the plastic bag, I gently blew air into it and then tied it securely. This helped to create space inside the bag so that the specimen is not crushed or pressed too early, and it also allows the plant parts to remain in good condition. The next step is pressing the specimens. The materials needed in this stage are kitchen towel, books, papers, cardboard, and specimen. First, prepare a flat surface using a piece of cardboard as the base. Then, place a layer of paper on top of the cardboard. This paper helps to absorb moisture from the plants. Next, carefully arrange the plant specimen on the paper. Make sure the leaves are spread out neatly so that their natural shape can be clearly seen after drying. After that, place another layer of kitchen paper on top of the specimen to cover it completely. Then, add another piece of cardboard on top to hold everything in place. Once the layers are arranged, press the specimen by placing a stack of heavy books on top. The weight helps to flatten the plants and remove moisture effectively. The next step is drying. Then, we need to place the pressed specimen in a warm, dry area, preferable under sunlight or in a well-ventilated space. This helps to speed up the drying process and prevents the specimens from developing mold. During the pressing and drying process, it is important to regularly check the condition of the specimen. We should observe whether the plant is drying properly and ensure that no parts are rotting or developing mold. If the papers become damp, they need to be replaced with dry ones to prevent moisture buildup. At this stage, the plant specimen is carefully examined to determine its identity. In this process, I use tools such as Google Lens to help recognize and identify the plant species. However, the identification should be checked carefully to ensure the accuracy by comparing key features of the leaf shape. After the plant has been identified, a label is prepared and attached to the specimen. This label contains important information including the common name, scientific name, family, and genus of the plant. In addition, details about the collection are also recorded, such as the place of origin, place of collection, date and time of collection, and the collector's name. Not only that, I also prepared the description and the picture of the specimen that I collected. The next step is mounting. The materials needed for mounting is glue, scissors, A3 papers, paintbrush, mounting board, and plastic wrapper.
In this step, the dry plant specimen is carefully attached to a herbarium sheet, and it is usually using a strong paper or mounting board. The specimen must be handled gently to avoid breaking or damaging its parts. First, the specimen is arranged neatly on the sheet. The leaves should be clearly displayed without too much overlapping so that all important features can be easily observed. Next, the specimen is fixed onto the sheet using glue. These materials help to hold the specimen securely in place while keeping it visible. After that, the label is attached to the corner of the herbarium sheet. This ensures that all important information about the specimen is clearly presented and easy to read.
The last step of making a herbarium is specimen care. After mounting, the herbarium specimen must be properly protected to ensure it remains in good condition for a long time. In this process, the specimen is carefully wrapped using a plastic wrapper. The plastic wrapper helps to protect the specimen from dust, moisture, and physical damage. It also prevents the specimen from tearing or becoming loose from the herbarium sheet. While wrapping, it is important to ensure that the plastic is clean, dry, and not too tight, so that it does not press or damage the specimen. The wrapped specimen should then be stored in a dry, cool place and need to be away from direct sunlight and humidity. The final product of this project is a complete herbarium specimen that is neatly prepared and scientifically organized. In conclusion, creating a herbarium is an important scientific activity that helps document plant diversity and deepen our understanding of plant biology. This herbarium not only demonstrates scientific organization, but also highlights the beauty and importance of ornamental plants in our environment. Thank you for watching.